Welcome, Edward. It's so great to have you here. And let's start with introduction to you. Where are you? Who are you? And what is your research?
I'm Eduard Chelebian. I'm doing my PhD here in Uppsala University. It's in Sweden. And my research is basically what we're going to talk about. So it's on the intersection of spatial transcriptomics and digital pathology to do analysis of cancer or basically that, I would say. Hmm?
Mhm. So, yes, this is exactly the title of our broadcast here, intersection between histopathology and spatially resolved gene expression. So, our audience knows exactly what histopathology is. This is pathology of tissue. We're looking at tissue, and the resolution that we get is we see the tissue, we can also see the cells, we can see the subcellular structures like nuclei and some organella. And we all know what gene expression is, but what is spatially resolved gene expression? Tell us about that.
I think the easiest way to understand is precisely going from what we know about gene expression. So I really like the metaphor they use when they presented the Nature Method of the Year in 2020, where they say that you have bulk RNA sequencing, and that's kind of making a smoothie. You have your tissue and you make a smoothie, so you don't really know if you have apple. You don't really see the apple, the banana. You only know that there's apples and bananas in your smoothie. So you know the cell types. That's the metaphor. Then you go one step further and you have single cell RNA sequencing, and that's kind of they say it's happening like a salad, like a fruit salad. So you can get your strawberry, your banana, and see the composition. So you can see the composition of individual cells. But to make single cell RNA sequencing, you need to dissociate the tissue. So you lose the spatial information. So here is where spatial transcriptomics comes in handy, which is exactly that. You have like a fruit tart, they say. So you exactly see where you have pieces of banana, where you have pieces of strawberry, and how these different fruits interact with each other and where they are in the tissue. So it's exactly that. You have the gene expression, but each of your rows, let's say in your matrix, have also two very important features, which is X and Y, which is the position of the spatial location.
hmm Okay, so I love your metaphor, by the way.
Not
But
mine, not mine.
um, okay, but what is the advantage of this in terms of science and in terms of like why is it important? Why do we want to need this? We have done the smoothie version of sequencing for so long and it has already had so many applications. Why do we go even more granular, single cell and in the tissue?
Mhm. So there's different way of doing this spatial transcriptomic, spatially resolved transcriptomic thing, and you can get either imaging based methods, which give you a subcellular level of the gene expression. So for each cell, you would have different spots that will tell you the gene expression, or you can go through the more sequencing based methods, which are more coarse in the resolution, but they allow to do a transcriptome wide, so you get the whole amount of genes in your experiment. And these two methods can be used, answering your question, for analyzing different things. So an interesting thing, for instance, is to see how tumor heterogeneity is expressed. So we know tumors are very heterogeneous, and sometimes it may not be enough to just put it on a smoothie and see that these mutations are there, and you would want to see, for instance, how the border between the tumor and the healthy tissue behaves, or which kind of mutations you have there. So it's very useful in general, I would say, to see interactions between different spatial domains, between different compartments, and also to discover different ones, different cell types that you were, or molecular profiles that you were not aware of because you were only looking at specific cells without seeing if this cell had many of these other cells around. So in those cases, I would say for heterogeneity and for spatial discovery.
Hmm. Okay, so question. You said that so it does not allow for profiling all the genes in the space where you're looking at, but rather you're targeting a specific gene and checking where it is. Do I understand it correctly?
So that's the main difference between the two methods.
Hmm.
The one that I said, when you get the subcellular resolution, there you can get in the order of hundreds of genes because you have to pre-select the genes, and in that case, you exactly, you have to design a panel of the genes you're interested in, and that's what you will then evaluate. But then
So this is very targeted. You know what you're looking. You know that there is a set of genes that are of interest. You're looking for subtype, some kind of I don't know, gene signature or some kind of combination in a certain location, and you're checking if it's there. Stuff from your panel is in this certain location, right?
exactly, and this family of spatially resolved gene expression techniques is usually called imaging based. So wherever you see the in situ hybridization or the fluorescent in situ hybridization, because FISH, it's how they what was the acronym. Those kinds of methods are exactly that. So they have the big advantage that you have subcellular resolution, but then on the other hand, you have this lower throughput. So that's one family, and of course that can help you in things, as you said, in subtyping. But then there's the other family where you don't need to select; it's not targeted. You can directly on top of your tissue, of your H&E tissue, and this is where what we will
Yeah,
we
tell
can
me about that.
exactly.
Because fish is something that the pathology community is pretty familiar with, and there are already diagnostic tests based on actually the cellular location of the genes that are being used for that. But you are telling me that you can do it on H&E? How can you do it on H&E?
Exactly. Yeah. Unfortunately, it's not me doing it. It's a very, very nice biochemist, and right now there's a lot of companies doing this. Maybe the biggest one and the biggest method here is Visium of 10x Genomics, where they, instead of doing this fluorescent imaging, where you are targeting these hundreds of genes, or this is advancing fast, so maybe now it's more than that. But in that order, what you do is you have in your H&E image, you do like small barcodes, like a matrix of barcodes, and then that's the barcodes that you will go and sequence. And thanks to having that barcoded,
Where is this barcode?
so you put the like a matrix of these probes that then will
Probes attached to something.
exactly,
To
that will
what do
capture
they attach?
so that's the mRNA.
Mhm.
So yeah, I really recommend you bring to your podcast someone that actually does it, not only analyze it like me. I hope
I
it's
will, and if you can recommend some people, I'm open to suggestion. Okay, so
absolutely
we have a just just to visualize the method. You have probes that attach to mRNA, and the
mm,
probes have some tails of barcodes of what?
exactly. So you have I don't know, you put a number to each of these spots of your matrix, and that's what you will use for capturing the transcriptomes. So in that sense, you don't need to specify the genes you're after, but each of these barcodes will capture everything that there is that they have there. So you will have tens of thousands of genes per spot, but you will lose, as I said, a lot of resolution here. So typically we're speaking 50 micrometers, something like that. So you will not have cellular resolution, but you will have a lot of genes to analyze.
Mm-hmm.
So as you say, the two families are kind of the opposite in that sense. Higher throughput with sequencing-based methods, but lower resolution. Lower throughput with imaging-based methods, but very high resolution.
Mm-hmm. And you are doing the imaging-based methods, right?
I'm doing the sequencing-based methods.
Sequencing, okay.
Exactly. The thing is, like, yeah, it's called sequencing-based methods, but they are the only ones that have H&E images attached to them.
Mm-hmm.
So you know exactly the position, and you can overlay it on top of your H&E image.
Mm-hmm.
So that allows you to see both the morphology and the expression very easily.
Okay.
And in the imaging-based methods, the only morphological imaging you have is DAPI staining, which shows you the location of the cells, which can be very useful. But of course, H&E is the golden standard for histopathology. So being able to have that is already widely used, and on top of that, having tens of thousands of genes, that as you can see, there's many possibilities of doing cool stuff there.
Mm-hmm. So you get, let's say, the from your collaborators, you get the H&E image and the overlay with all the genes in the, I don't know, is it square? How do you choose your the part of the slide, pathology slide, where you want to analyze it? Do you go in and check where the invasive margin of the tumor is, or what? How do you select your regions of interest? How big can your region of interest be per, I don't know, question? And our third question, like, can you do it on a whole slide? And does it make sense?
Mm-hmm. So as for choosing your region of interest, of course, you're trying to choose a region that you morphologically have seen that has different kinds of tissue on it, because you want to address heterogeneity, right? We said that's the most useful thing of having the spatially resolved gene expression. So imagine you have different different subtypes of tumor in your tissue. You have a little bit of stroma, you have a little bit of benign tissue. So you're interested to see what differences arise there. And in the case of that's how you choose the region of interest. And I would say the arrays they use are around 4 by 4 millimeters. So they can be
That's pretty
on,
big.
yeah, that's fairly big, I would say. And it can be like a biopsy, for instance, a cut of a biopsy, of course. And that's exactly what many people are doing, biopsying, for instance, a prostate, and then you get this pretty big chunks of tissue. You select where you have interesting interactions between tumor and stroma and benign tissue, and that's what you send to then sequence and get the spatial transcriptomics experiments done on.
Mhm. So I assume the region is selected by a pathologist or with pathologist assistance, and then you get the both the image and the overlay from the gene expression. What do you do with it? Like, how do you deal with it?
Yeah, now is where the fun starts. These things before as I said, I recommend you to get some clinician that, or someone in the laboratory that
So
really knows.
just to set the like the collaboration that you're part of. You work with a pathologist. You work with the gene expression team, or who's in that team, and
Mhm.
then you're on the image analysis or the i- image to gene expression analysis part. That's your task, right?
Exactly.
Okay, tell
That's
me
my
all
task.
about it.
And yeah, here I can speak more freely in that sense. So right now you have your image, and you have these spots that are have different gene expression. So you can analyze them separately. First, for instance, you can get the gene expression and see where do you have genes that are expressed in a similar manner. So as I was saying, if you have here a lot of strawberries and apples, you can say that this is a genetic region, or a domain that has very homogeneous gene expression. You can, and that you can do only with the gene expression. Or you can do that also what is called segmentation, right? On your normal tissue, on your histopathology, in your H&E slide, you can just do exactly that. And one thing we did was to take these two things done in an unsupervised manner and see how they correlate. So, of course, the phenotype, which is the morphology here in our case, is determined by the genotype, which is the spatial transcriptomics in our case. So we wanted to really see if, for instance, features extracted by a convolutional neural network on the H&E image had like a similar profile as some genes. So I don't know if you.
So cool. I understand. I say this one I understand.
Yeah.
I've been challenged by your group today regarding like how very much I follow what you're saying, but this actually I'm following. So, but just to translate it into simple language, you kind of cluster where the similar genes are expressed in the gene part of your mask, right? Then you map it on, and then you have this information. Then you have a convolutional neural network that works with the image. So here I have a question, like how do you know what features it is actually producing? Is it a segmentation mask? What kind of activity or what kind of task do you have for the convolutional neural network that works with the image?
Exactly. This is a very active field, I would say. So I guess you already know that usually people use ImageNet for feature extraction. So what you do is you feed a network, you feed an image into a network that is pre-trained on ImageNet dataset, which is a natural image dataset, so cats and dogs and houses. And then you go all the way up to the penultimate layer. So before the prediction is made, whether it's a cat or a dog, and those features that are used for that, you take them, and that's what you consider your morphological features.
Okay, so you can basically extract that before you get to the prediction.
Exactly. That's the layer you're using. So you're transforming your image into a bunch of features. So ImageNet may not be the perfect data set to pre-train your network on. So right now we're looking into self-supervised approaches that will learn from the data, maybe more interesting things without any label, of course, but trying to learn the natural, let's say, structures of the data, and then you extract features with that. And yeah, I guess some of the people at Radboud will speak about this because they know about that. They know a lot about that, but basically you would get these masks, and as you're dividing your image into small patches, each of these patches will.
One question: masks of the features, not of the prediction, right?
Not of the predictions,
Okay,
exactly.
so you have mask of the features on top of H&E, and then you check if your genetic clustering corresponds to any of the features.
Exactly.
Okay.
And that was pretty fun to see, because you see that even though because we have the annotations also, but we're not using them, we're leaving them for the end, and we see that even though some of one region is annotated as cancer, let's say, or as a specific subtype of cancer, you see that the gene expression here is different than the gene expression in this part of this. And we also saw that there are some features that morphologically are different here than here. So that was pretty fun to see how this connection between genotype and phenotype is really there. And in the end, the categories we're using are we have to put a limit to the categories when we say a subtype that can be a subtype of subtypes, and you could go infinitely there. So that is a really interesting thing you can do. And of course, some groups they are doing, they're saying, okay, so there's a lot of information that is correlated between the gene expression and the H&E. So why not fill the gaps of our gene expression that we just said that it doesn't have perfect resolution. Why not use our H&E to guide the gene expression and fill the gaps, let's say, between the spots, right?
Hmm.
So that's something some groups are doing, and to create like a super resolution gene expression or
Question, because something that has a huge potential and is pretty. There's a big hype about it in the diagnostic pathology community, where you can predict the molecular uh, signature or molecular properties of tissue from an H&E image, and it's usually your label for the image would be then the result of the molecular test, but you have like no idea where this information comes from. And even if it very much corresponds and has very high accuracy, there is no way in looking or verifying is it coming from where it actually needs to come. Would what you're doing be something to add explainability aspect to that type of testing?
exactly that would be a fun thing to do. I think I've come across some studies doing exactly that. So they try to predict the gene expression or the what you were saying, have a like a specific label, and then see if the places where the attention is or the attention of the network. So these places, the network said, okay, I'm interested here. I'm interested here. Do those places correspond then to the spatial distribution of that?
See, now you're filling all the gaps. I was like, attention, okay, but if there is something that you cannot visually verify, then you don't know if this attention makes sense. And here you'll add another piece of information, then can add up. Fantastic.
That would be a very fun thing to do in large scale. Of course, I have to jump in and say disadvantage of this. You would usually these experiments where you predict the single cell, the bulk RNA, or these things, you have a lot of images, and doing this spatial resolved gene expression for a lot of images would cost a lot of money. So
Yeah. Do you
you would
know
have them.
what the cost, let's say, cost per experiment, the experiments that you're doing, what's the, how many images are you working with to make your conclusion?
Mm.
What is the number that is like the minimum number to, what's the cost of it? Because so how I see it, is okay, there is something very promising where you can, uh, there is a potential to skip a molecular test because the prediction from H&E is so good that we are like, it's always working. But we need to add this level of explainability once or twice to like justify the use and the switch. Um, that for me would justify the cost. But tell me what the cost is and what are those experiments with how many images, how much tissue?
So I would say good news and bad news here. The good news are that I think the gene expression in general, the genetic community is very open and sharing in that sense, and there's a lot of data that they share. And I think the spatial genetics or spatial transcriptomics community also carried on with that, and almost every study you see, then they attach in the supplementary some kind of data that you can yourself work on. So that is pretty good news. But then the bad news is that usually this kind of papers use one patient and 5 slides of the patient. That's kind of uh, a,
Okay.
a good experiment, a good paper, or yeah. So it's one patient and a couple of slides of the patient, because it's really hard to do, and really costly. Apparently, I couldn't say a number here, but yeah, for what I've said, for instance, it was just 7 slides of one patient, or
Hmm.
of one prostate was, and that was previously published, and it was already available for people to use. So you see the good thing and the bad thing in one in one paper at the same time. So
Yeah, so definitely this. So these are kind of like proof of concepts that the method, and like figuring out the method, definitely for justifying a major switch in the diagnostic testing. That we need to go a little bit higher on the number of patients, and it has to be done at scale. But at least the methods are there, and if then at the end you can have a return on investment that justifies this investment, then it's totally worth it.
mm.
So, okay, we have 5 more minutes before we go. Tell me, tell me the details of your experiment, of this prostate experiment. Can you already disclose it, or are we waiting for a paper to be published?
No, this was already published.
Okay.
So it was using an already published data set. So that was the sharing part. I was saying that was very nice to be able to work with this data. And what we basically did there was as I said to get let's say profiles of gene expression from our spatial transcriptomic experiment. So each of these profiles would like group all these tens of thousands of genes into, I don't know, 10 or 20 profiles, which is more manageable. Because of course, when you have 30,000 genes and then you have thousands of features, deep features, and then you try to correlate those two, you're going to find correlations everywhere because that's huge amounts of data to work with. So we have to try to do the meaningful correlations. Once you get
know which ones are meaningful.
Exactly. That's the idea. So by defining these profiles, you at least know that what you're trying to correlate is something that happens very often, and some regions that have let's say a gene expression that means something, and then you try to correlate with that, and you're sure at least that that what you're finding are not just spurious correlations. It's something that is genetically relevant. And then the pathology workflow, the digital pathology workflow, was very similar to what other people do, in the sense that you divide your huge image into patches. And something very nice you can do here is, because you have these spots on your tissue, you can just take the center of your patch in this spot that has the gene expression, and that very easily you can assign the same position a morphological let's say feature, or morphological vector, and then the genetic vector. So you can imagine there's many people that have been doing recently interesting stuff with that, because there's very nice for integration, right?
Mm-hmm.
So exactly, when you divide your image into patches, then you can feed it into a convolutional neural network and get your features, and that's how we did the correlation in the end.
So do you have like a certain feature that corresponds to a certain gene?
Exactly, that was what we found in the end, that the features that were extracted, and then by doing some simple unsupervised clustering of these features and seeing what kind of morphological clusters we get in our tissue, there were many correlations in between those two things. So that's basically how we did it.
Okay.
Mhm.
Thank you so much for letting us know about this. I learned a lot as well. I learned about the huge potential that's out there, and how we could explain stuff that's not yet explainable.
Mhm.
Thank you so much for joining me.
Thank you for